Well, thank you everyone for joining us today. For those of you that were able to catch my proposal presentation yesterday, the first half of this will be a little bit of a repeat, including some recycled photos, but the results should get more interesting. So the title of my presentation is Increasing Rocket Box Use by Maternal Indiana Bats in Northern Kentucky. And this included efforts from both the Dodds and the O'Keefe labs. So first, to give you an overview about bat boxes, they're being increasingly used as mitigation tools for all different types of bats for various reasons. And there are many different styles of bat boxes. As you can see here, we have the more traditional style, but they can range from shape and size and material. Now, Indiana bats, specifically, research has shown that they prefer rocket box style bat boxes, and that is this one right over here on the right. Now, our research needs, we know that Indiana bats prefer these rocket box style boxes, but we don't really know about the preference for different designs of that rocket box. So you can make different modifications of different designs, right? And then we also want to know how these bats acclimate to these rocket boxes over time. And that has led to our research objective, which is evaluating the maternal Indiana bat use of varied box designs across varied landscapes and over multiple years. Our field site is Veterans Memorial Wildlife Management Area. It's owned and managed by KDFWR. And on the left here, you can see this is just the overall view of the site. It is in Scott County, Kentucky. The map on the right, that star is where Scott County is. I think it's about 2 counties north of us here in Madison County. But this is the overall Indiana bat range. And as you can see, it is right smack dab in the middle. So this is a great place to study this species. Our site is a little over 1,000 hectares. It's part of the Hills of the Bluegrass ecoregion. So it has various hills and ridges, oak, hickory, and cedar trees. There's also a good amount of mesic forest and streams here, as well as a few pond sources. The first part of our methods were Reed Crawford built various rocket boxes, a large number of rocket boxes. Here is the reference design. You can see a rocket box is basically just a tall, narrow box, and this one has 2 chambers and a vent right here that allows heat to escape so it doesn't overheat. And then Reed also built 4 different other box designs that were designed to either increase or decrease that overall maximum box temperature. And the first of these is the vent removal box. So it is just this reference box design, but with that vent gone, and that will increase the overall roost temperature. The second, sorry about that. The second design is the white tile roof, which is the same reference, but with a white roof. And that's to kind of reflect some of the solar radiation, particularly near the top of the box, because that is where they get hottest. And then we have the chimney design, which is this box with a large, or a very tall black chimney on the top. And you'll see a picture of that on my next slide. And then the last box type was the external water jacket box. And basically this outer chamber was closed off and inside. And so water heats up and cools down slower than the air temperature, right? So it was kind of there to buffer temperature changes and keep it at an overall kind of a steady temperature. Those did have to be removed after the 2019 field season, unfortunately, due to some structural changes, but we did get 1 season's worth of data out of those. Next, we had to install the boxes. So there were 20 rocket boxes total. There's now 16 because those water jackets were removed, but they were installed in clusters of 5 across 4 different solar treatments. You can see here on the right what those clusters looked like, and you can see that chimney on that chimney box right there. And so for this map, our open cluster is in this white area, which is basically just kind of open space, generally fields here because it is a managed area. And so it received predominantly full solar exposure. We have our east cluster here, which was along a tree edge, and it received predominantly eastern sun, while this west cluster, which is also on a forest edge, received predominantly western sun. And the forest cluster here was in the forest interior, receiving far less solar exposure. In order to monitor these roosts, we did spotlight checks and exit counts 3 to 4 days a week throughout 2019 and 2020, the summer seasons. You can see here Reed is spotlight checking the bat. And we do this to get an estimate of the number of bats in there. We also record species and if any of them are banded. And in this picture, you can see that we can differentiate between the species while looking at these roosts. These are Indiana bats up here, and this is a big brown bat down here. So the Indiana bats have this more narrow muzzle, and it tends to get pinkish on the end, whereas these big brown bats, in addition to being much larger, which you can't tell in this picture, but we generally can see that, they have a much darker brown muzzle or almost black. It's wider, and they have these distinctive lumpy glands there that make them easy to tell apart. And then for exit counts, that is just to get the overall abundance within, or a number of bats that are occupying each box at a specific time. For our results, here we have the mean daily count per rocket box, and we have divided that up between solar treatment and then also reproductive condition. So for solar treatment here on the left, you can see that, or actually both graphs, you can see there's a huge increase in number of bats using these boxes from 2019 to 2020. I think it's nearly quadrupled the number of bats. So that's great. They are finding these boxes, and you can see the overall trends seem to be similar. They're primarily using these east and west cluster boxes. The forest cluster had minimal use, and I did not include the open cluster in this graph because we'd never counted any bats in those boxes. And then for reproductive condition, you see that it's also a very similar trend going from pregnant to lactating to post-lactation. As the season progresses, it seems that these bats are starting to use our roosts more and more. We also just looked at the total count of bats in all boxes per month. And here you can see we did that again for these solar treatments. And it seems that, as our last graph showed, they do prefer the eastern and western boxes across both years, but it doesn't look like there's any sort of specific seasonality there. They'll use them during all parts of the summer season, with the exception of this forest cluster. You can see in 2019, it was only used in May. It was used a few more months in 2020, but it was still the primary use is in May. And this is generally before the maternity colony forms. So it seems as before the maternity colony forms, they like those forest roosts. And then as it progresses, they switch to the east and west clusters. And my last set of graphs here is also the total count per month for all the bat boxes across 2019 and 2020, but this time we're looking at specific rocket box designs. In 2019, there really doesn't appear to be any specific preference here. It's a little bit all over the place. And in 2020, you can see that external water jacket box was removed. We see a little more of a pronounced pattern. It looks like they are using the chimney and the vent removal roosts the most out of the other designs. Now, we haven't run any statistical analyses across both years yet, so we can't say for certain whether this is actually statistically significant, but visually we can see that they are using these boxes the most. Our overall conclusions are that we believe the increased box use from 2019 to 2020 is likely due to just the time it takes for bats to discover new roosts. But other explanations could be bats are developing search images for these roosts, and they could also be assessing microclimates of these roosts through trial and error. We are still in the works for getting some of this published. Reed Crawford should hopefully have a publication coming in the near future looking at all of this data, so I'm looking forward to seeing that. We would like to thank the KDFWR for supporting this project, and also the U.S. Fish and Wildlife Service for funding this. I would like to thank all of the members of both the Dodd and O'Keefe lab groups, and those will include Reed Crawford, Dr. O'Keefe, Dr. Dodd, Evan Mosier, Elizabeth Robinson, Dalton Bundy, and JP Lyons. We'd also like to thank the EKU Graduate School and the Department of Biological Sciences. These are my references. And does anyone have any questions?
So Shannon, I've got a question. Do you all have any sense as to, so you said that these rocket boxes are kind of preferred by the bats over other structures. You guys have any sense why that is so? Is it a matter that, why is that? Do you,
yeah, so There is a theory behind this supported by a few different studies, and that is since these boxes, they're designed, they're narrow and they're very tall. And so studies have shown that bats like roosts that are very tall because they have more variation in their temperature. And so that way they can kind of move throughout the box as temperatures shift throughout the day to find their ideal temperature and microclimate. Whereas if a bat box is very short and it gets too hot, then the bats, they either have to leave or they're just stuck there. Whereas if they have a very tall, long box, they can kind of move out and decide where they want to be throughout the day as the temperatures shift inside.
Okay, so follow-up question, if I may.
Mhm.
Compared to other mammals, so do bats have a much narrower temperature tolerance range, shall we say? It seems like they get rather uncomfortable and they want to move around if it's a few degrees difference. Is that typical of many mammals, or is it just something that's very bat specific?
I don't actually know the answer to that. I'm not sure how other mammals are able to tolerate those temperatures. I know specifically here we focused on maternal bats, and maternal bats, it's very important for them to have high temperatures because research has shown that higher temperatures increase the rate of pup development, whereas lower temperatures will decrease that rate. So they want these warm temperatures, but if it gets too hot, which for Myotis species, that threshold is 40 degrees Celsius, which these boxes can get over that. That's when they start to exhibit heat stress. And so it's kind of a balance of finding a nice warm place for them to raise their babies, but that's not going to get too hot that it's actually going to harm them.
Cool. Okay. Thank you, Shannon. Thank you. And folks, if you have questions for in the seminar series, just holler out, or if you want to post a question in the chat box, I'll keep an eye on that too.
Okay, I have a question, Shannon.
Yeah.
So it seems like, most of the designs for these boxes are sort of designs that will either impact, perhaps temperature, or I'm guessing like gas exchange in the case of the venting. So a question I have is, have any studies been done to kind of figure out with these various designs, the degree to which the temperature or gas exchange would be affected by those designs? Is it better to have a reflective strip on the top, to keep temperatures down, or a vent, do you have kind of any sense of that?
Yeah, so the first part of Reed Crawford's thesis was actually assessing the microclimates of these boxes. So in 2019, we did have some eye buttons in there that were recording temperatures in the different parts of the roost. And so we were able to record like the minimum and maximum and average temperatures of these roosts. And I think as far as these specific designs go, the vent removal obviously got the hottest. And the average temperature or the most stable roosts were those water jacket boxes. But again, they unfortunately had to be removed. And I think they're working on redesigning those to make them more long-term. So yeah, we do have some research on that.
Yeah, and I'm not necessarily familiar with anything on the ground like gas exchange. Are you, Shannon, with what you've read?
Yeah.
Yeah, I mean, that's something that people talk about, but we don't have an understanding on it just yet. Definitely like a lab-based study that would be cool to kind of think about it in that kind of context.
Yeah, cool. Thank you.
Shannon, is it fairly usual that bats get together in different species in a roost?
Not really. So in our boxes, the only two species we've ever found have been Indiana bats and then those big brown bats. The big brown bats are only in there occasionally, and if they are in there, it's usually only one or two at a time, and they're almost always in a different chamber. So they're kind of roosting, outside. I think once I did find one big brown bat roosting in with the Indiana bats, but that's highly unusual, and I'm sure they weren't very happy about that.
I've got kind of a practical question. I've had a bat house in my yard for 3 years and it's never been used. So are these kind of bat houses actually available commercially to try to use, or is it something that you just have to really sit down and design them and make them yourself?
I'm not sure about that. I know Reed obviously built these himself, and there are some schematics out there. I don't know if anyone sells these specific types of boxes commercially. But what I do know is that a lot of it depends on placement of the box. In addition, design placement is key. And then also, the bats took up artificial roosts here very quickly because they were documented there. We knew there was a colony there. Whereas if you haven't really seen bats near you, it might take longer for bats to actually go out and find them.
Yeah, I mean, every day if I'm out in my yard, I see them flying. So they're in the area, but they just have not used the house.
Pat, can you make it so that I can share a screen?
Yes, 1 second, Luke. Yes. Luke, you're a co-host, Luke.
Awesome. So like the bat box design that like this sort of this is an original, this may be the original bat house bat box design that people have sort of worked off of, but John McGregor with Kentucky Division of Fish and Department of Wildlife Resources, and then Dan Dorson designed this, and this is actually on the EKU biology webpage. There's like a variety of old like 90s bat stuff that's sort of tucked away somewhere on the webpage. But like this is like a 4x4 that you chainsaw some grooves into, and you basically slide like the 1x6s like over the top. And so this is like the OG original kind of design. And so you can build these types of rock boxes. But the bottom line with like bat houses is the higher you can get them up, the better. The closer you can get them sort of like to an important feature. And don't put them on the side of a barn or the side of a house. So get your bat box about 20 feet up off the ground. Don't put it on the side of your house, and put it sort of in a relatively open area, but near an edge. Then make sure it's painted the appropriate color. Sort of drab brown works best here in Kentucky to make sure it's going to get hot. The biggest issue with bat boxes today, like that people put out, is that they don't get hot enough. Like that's the general statement.
Interesting.
In addition to that, it might be helpful if you're able to figure out what species you have, because if it turns out you have eastern red bats, then they don't roost in boxes. They just like hang on to trees and pretend to be dead leaves. So if the red bats, you're just not gonna get them in a box ever.
Thank you.
Any further questions for Shannon? Hearing none, Shannon, thank you. That was a really great job, I think, and a nice follow-up to your thesis proposal presentation yesterday. So congratulations to Luke also for having such a fine set of students working in his lab. Very, very impressive. Okay, let's move on now. We'll have Miss Sarah Baker as our next speaker coming up. Sarah's also a member of Luke's lab, and she has done some work in the museum here. And she'll be talking about in the Mammalogy Museum, I believe, and she'll be talking about that work then. So Sarah, you should be able to screen share also, I believe. If you want to pull your presentation up, there we go. Yeah, we got it.
All right, great. So to reiterate Shannon's statement, thank you everyone for tuning in to hear us give these talks and hear me talk a little bit more about what we have in the EKU mammal collections. I've been the assistant curator of the mammal collection since last semester, so I've kind of had the really great opportunity to work in the collection as part of my service assignment, as well as part of my thesis work, which those of you who tuned in to my defense last week, some of this may overlap a little bit, but a lot of it will just be kind of looking a little bit more in depth at some of the records that I alluded to. So to give you guys a little more general overview, I have these two figures from Carson McNamara's poster she presented at the Southeastern Bat Diversity Network meeting. And so the first figure on the left shows all the museums that are located across the US. So there's at least one natural history museum in each state within the US. And These are incredibly important for documenting biodiversity, for long-term studies of species richness and species distributions, as well as providing records for taxonomic studies. And there are over 2 million specimens cataloged overall across the US. Within our collections here at EKU, we have records from 4 countries. So we have records from Mexico, Canada, Sweden, and 18 states within the US. And of those records that we have within the US, the vast majority of them are from Kentucky. And we have 1,600 specimens overall, and most of those are from the Rodentia. And I'm going to talk quite a bit about the Rodentia today, as well as the Soricidae. There's another figure on the left from Carson's poster, and this is showing a little bit more of the timeline of the EKU Museum, but it was first started in 1955 with records of white-footed mice. And since then, records have been kind of added up until 2007, until we have our total number of records. And on the right here, I just have a plot of the number of records that have been added that are specifically small mammal records. So these are all the Rodentia and Soricidae records within the collection. And you can see as there's not really much of a trend overall. There's been a lot of variation in the number of records that have been added across the years. But important to note is that really since 1999, only 2 records have been added in 2007. Other than that, there haven't been any records really added to the museum in the last 20 years. And to break this down just a little bit more, I created these 2 plots looking specifically at the Rodentia in plot 1 and the Soricidae in plot B. And so in plot A, you can see that this plot's really similar to the one on the last slide. And that's really because most of our small mammal records from Kentucky are Rodentia, and these plots are specifically from Kentucky. I think I forgot to mention that on the last page, but you can see again, there's a lot of variation in the number of records that have been added, and just 2 records that have been added since 1999. So not a whole lot of updating has been conducted in those Rodentia records. And for our Soricidae, you can see that we have a little bit more of a trend here. There's kind of been this increase in the number of records that have been collected for Soricids, but this is stopped at 1999, and we haven't had any new records since that point. And so I mentioned that this ties in quite a bit to my thesis, specifically my 3rd objective. So I'm going to be assessing status of specifically reclusive small mammal records within the EKU Natural History Collection to really get an idea of how many species we have recorded in the collections and how many records we really have for each, to get a good idea of what our kind of overall documentation is of the biodiversity within the state. And the reason why I want to focus on these really reclusive small mammal species is because we don't really know quite as much about them because they're quite difficult to trap. So this slide is probably going to look very familiar if you tuned into my defense, but I just want to reiterate this point, looking specifically at Sorus longirostrus, the southeastern shrew, and Cryptosis parva, the least shrew. These trapping success rates from Magdock and Guyan are specifically from pitfall traps, which are actually the best way to catch shrews. And you can still see that there's a really low trap success with only about 1 individual per 100 trap night. And this really low trap success leads to us not having a lot of information on these really interesting species. And I was really curious to see exactly how this would be represented within the collections, if we would see gaps, because small mammal species can be difficult to trap and get records for. And so this first map that I have here, I created this in R using the number of records that we have within our museum database. And I did that by species and by genus, and by county. And so you can kind of see right off the bat that we have a huge concentration of these records in Madison County. So we have about 400 small mammal records from Kentucky, and over 300 of those were collected in Madison County overall. And in all the other counties that we have data from, so these light blue counties, we have less than, we have either 10 or less than 10 records. And all of the gray counties that you see, we don't have any data for. And so I wanted to break these down a little bit more just to kind of get a closer look at specific groups, starting with the Rodentia. And what I found is that we have 10 genera documented within the collections. And of those, you can see a kind of a similar distribution on the map that we saw on the last one. So we have 358 Rodentia records, and 239 of those are were collected from Madison County. But we also see a slightly different kind of bias looking at it this way. A lot of the records, 154 exactly, were from Peromyscus, and 55 were from Microtus. So we're also seeing really strong bias towards specific species within the collection. And to look a little bit closer at the Muridae, which we have 2 genera up within the museum. You can see, again, even as we break it down even smaller, this really similar pattern of having this strong bias towards records within Madison County, as well as towards species. So we have 104 total records for murids, and 50 of them are from Madison County, and 70 of those total records are from the Mus genera alone. I also want to look specifically at the Soricidae. So we have 3 genera of Soricidae within the record, within the collections. And within this group, we see the same exact bias, where we have 126 total records, and 65 of them were collected in Madison County. And we also see a particularly strong bias towards the Blorina genera, with 72 records from that genus alone. And I also wanted to look specifically at the number of species. So last 3 maps, just looking at the total number of records, and this is breaking it down by the specific number of species that have been recorded within each county. And so for the Rodentia overall, I found that most of the genera, you can see, only had 1 species represented within the collections, with Microtus, Neotoma, and Peromyscus being exceptions to this rule. But even though we're looking at a slightly different classification here, we still see the same pattern of the collections being biased towards Madison County. So we have 12 records from Madison County, and we had around 3 records here from Harlan and Letcher County, and some of these other more central counties as well. And then all the rest of them only had 1 species recorded within the county. And looking more at the murids, you can't really see as much of a pattern here because we only had 2 species, Mus musculus, which is the house mouse, and Rattus norvegicus, the Norwegian rat. So you can't really pick up too much of a pattern here, but you can see that we did find both of the species within Madison County, and again, kind of finding just 1 species here and there throughout the other counties that we have records for. For our Soricidae, we have 5 species in total. So we have Blorina brevicata, our northern short-tailed shrews, Cryptosis parva, the least shrew, and then we have Sorix cineris, masked shrew, Sorix hoyi, the pygmy shrew, and then Sorix longirostris, which I already mentioned, the southeastern shrew. And again, we see a little bit more of that bias towards Madison County. So we have 4 of those species found within Madison County alone. We had 2 found within Harlan County, and then 1 throughout the rest of the county that we have records for. So again, just this continual pattern of having these different biases in the data set. And sorry, here we go. I also wanted to create a map that gave an approximate actual species richness distribution across the state. So I pulled these numbers from Barber and Davis's Mammals of Kentucky book, and I essentially did something very similar to what I did with the records, where I went through and tallied up for each county the number of species that were either had records within that county or were expected to be present within that county based on the maps that were provided in the book. And what I found overall was that, 1, we do have species throughout all counties in the state, so we're definitely missing some records within the EKU collection, but also we see those kind of centers of biodiversity quite differently. So Rowan County and Callaway County become those kind of centers of species richness with 13 species each. And then other counties like Harlan and Pulaski have 12 each. And so we're not really seeing that bias towards Madison County when we break it down more like this, but we also see just a lot more variation in species richness by county than we can really observe in the other maps that are based on our collections. And we also have quite a few more genera that are known to be present within the state than we have in the collection. So the lists that I have on the right of the screen here are from the Barber and Davis's book. And see that we have quite a few of these genera within our collections, but we are missing the Andadra or Azamis and the Napiozapis. Those are all records that we're missing. We're also missing Parascalpus. And I didn't really talk about the Televidae too much because I was focusing quite closely on the Soricidae and the Rodentia, but we only have 2 records of the Scalopus in our collections, and they're both the same species, Scalopus aquaticus. So again, it's kind of another area where we are lacking a little bit of data. And this also isn't a fully comprehensive list of species. The Barber and Davis book did not include Soricus bar, which is a very uncommon species, but I do know from records provided by John McGregor that they have been trapped in the far southeastern part of the state in Harlan County. And so there are still species that might not be included in all species lists that we have documenting biodiversity in Kentucky. And so being able to kind of expand our records and trap more broadly will kind of help us to get a better idea of exactly what species we have present in the state and what their distributions are. And so overall, what I found is that we haven't had any large additions to the collection in recent years, with only 2 being added in 2007 to our Rodentia collections and none being added to our Soricidae collections since 1999. And I also found quite a few of those biases within the data set. So most of our records collected from Kentucky are from Madison County, which does make sense since the collection is located here, but which does leave a lot of gaps within our knowledge of species distributions within the state. And again, most of these records that we've collected are from kind of these 4 large groups, Peromyscus, Moesplerina, and Microtus. And the reason why these are important is because developing a very comprehensive record of species within the state, and one that's recent and current, is going to allow us to conduct long-term studies of species distributions within the state. And it will also create records that can be used to develop site or species specific management plans, especially for species like the red-backed vole that we know are present in Kentucky and that are of special concern. So I just want to take a minute to acknowledge some individuals and the Office of Kentucky Nature Preserves, EKU Graduate School Department of Biological Sciences, who provided the funding so that I can work on this project. I also want to recognize some specific individuals, Carson McNamara, Courtney Hayes, and Reed Crawford, who are all former museum assistant curators and who conducted a lot of work updating and maintaining database records and conducting their own status assessments of records within the Mammal Museum. And also my advisor, Dr. Dodd, and fellow lab mate Shannon Howe for all of their guidance and support through the development of this project, as well as John McGregor, who provided records on Harlan County and some of the species that I don't have any other records of. And if you'd have any questions, I would love to answer them now.
Thank you, Sarah. Great presentation. Questions, folks, for Sarah?
I had one.
Please.
I was just wondering if you've looked towards other museums to see what their records are through Kentucky, like through GBIF or one of the Symbiota portals. So how well is Kentucky represented in other museums, I guess is my question.
No, it's a great question. And it's funny that you mentioned GBIF because I've spent a lot of time creating maps of Rodentia and Soricidae maps with GBIF. But interestingly, what I found is that we just don't have any like state data. I put in all of the species from the Barber and Davis book with a limit of 300 records each, and I got maybe 20 points within the state. There just really wasn't much to work with. And there are definitely points throughout the rest of the country, but it didn't really give a good idea of what we know is present within the state or the distributions based on other records that I've looked at. So that really gave me the impression that that particular source, like also there's just not a lot of data like present on these species. And for anyone who's not very familiar with GBIF, it's this database of records of species occurrences from literature, fossil records, museums, and they do have, I don't actually know the exact number, but I know it's like well over 2 million records at this point. So I was really surprised that I wasn't able to get a lot of data from that particular resource. I would really like to look into the records that are in other state museums like the University of Kentucky, and I was recently told another museum in the state that may have some records that are site specific to where I'm going to be working in the summer, but I haven't specifically looked at those yet.
So Sarah, tell them a little bit more about that Redbird data set that you've had a chance to look at from Kaiser.
Oh, absolutely. I didn't pull that into this presentation because it is very specific to Harlem, Letcher, Knox, Leslie County, Bell County, and all of those. But those were all records, and so I had the Redbird district records, which showed species that have been trapped across those kind of counties, and then I also have management plan data, and it aligned fairly similarly in the number of species that I found, although they have found Sorix fumius, the smoky shrews, within those counties, which we don't have within our collections. But for the most part, they have some very similar species that we've trapped, but they also have records for Letcher County, which we don't really have represented well within our collections.
Yeah, I think that the Redbird inventory, I think what James Kaiser told us is that that actually is on hand at Moorhead. So whenever he mentioned that collection at Moorhead University, I think it's the samples from that. And is that from like 91 or 92? Is that right?
Yeah, it's right in there. I think it's 91.
Here's my other question. Is there plans to make this available? I just pulled up the collection, the Symbiota portal for small vertebrates collection, and it looks like our fish collection and herp collection are listed there. So you guys have plans on making the data public?
I don't know of any plans currently. I think that would be great if we could make it public, but I have not been plans to do that.
Now we have an enthusiastic collections specialist working on her thesis. It's definitely a direction to take things.
I'll share with you all a rather interesting story from Genter Schuster, David, regarding collections, okay, and Sarah, how important they are. So I guess it was back in the 90s, back in Memorial Science, my office was across from Ganter's. One day he got a phone call from Ohio Department of Transportation. Hey, we got a bunch of jars of stuff here with things in them. We're going to throw them away. Do you want them? And it was literally, I think, David, the best collection of Ohio aquatic invertebrates around. They were going to throw them out in a dumpster. So luckily they knew about Genter and called him, and he drove up there, I believe, and got the entire collection, brought it down here. So, you know, if we don't look after these materials, they're lost forever. And here in Kentucky, especially, as David and Dr. Harold can tell us, some of these things are now extinct from their various localities. So these collections serve as amazing historical records in terms of where species once occurred. And very often people think, oh, it's just a bunch of dead stuff, just throw it out. So I think, Sarah, your efforts are important to sort of point out to folks the value of these collections. It's not just a bunch of dead stuff, but they're historical records. They can help in terms of biogeographical studies. And Ganter's story, when he was just astounded, he said, it's an amazing collection. It was the best collection of aquatic inverts from Ohio, and we almost lost it. So now we have that actually in our collection in an invert museum. I'm also going to post, folks, if I get this right, yeah, in the chat room, University of Michigan, Brad Ruffel, a colleague of ours who is now up there, is hosting a symposium starting today on natural history collections. It'll go for 5 weeks from approximately 1 o'clock to 4 o'clock. If you go to that link, you can actually sign up. It's free, but it'll be a series of discussions about the value of natural history collections like ours, for example, and how they're being utilized for various scholarship and efforts. One challenge we have, Sarah, is always convince people of the value of these particular collections. I've been involved with the herbarium at Tennessee and also at Michigan and here, and so we have to constantly tell folks why they're so important and also keep on adding to them to increase their value. So thank you for highlighting the importance of our particular mammal collection.
Sarah, I have a quick question. I bet a student in mammalogy could answer this, and I've never studied mammals formally, but could you explain like the trends in species richness in the state? I know for plants, it's highly based on physiographic region and just the topography, but what is it for mammals? I couldn't really pick out a trend in the maps.
So I haven't taken mammalogy myself yet, but based on the reading that I've done preparing for my thesis, I would say a lot of it is based on specific microhabitat characteristics. A lot of the papers that I've read have stressed the importance of presence of coarse woody debris, providing habitat for species, but a lot of it can be very species specific. So I talked a little bit about Sorix dispar, which are found in the far southeastern part of the state, and those are really only found in rocky habitats. You're not really going to find them anywhere else, which is why they're found in those mountains in the far southeast. And you also have species like Permiscus leucopus that are, habitat generalists, and those were really found just across the state as a whole. They're found in every single state. And then there's some species like the southern short-tailed shrew, which are only found in the far southwestern part of the state, but from reading the literature, it doesn't seem like there's really a specific reason as to why, at least that anyone's really determined yet. So I think it's really very species specific to microhabitat characteristics. And I think that's the best answer I can kind of give you at this point.
No, thank you. That's super interesting.
Hey, Sarah, as you've been looking through the specimens that we have you figured out how we've acquired them? So part of the distribution may well be that we had somebody that was working in such and such a county, but not working in the county next to it. Or are these generated by thesis projects, or how have we come by all these specimens?
Most of them are by targeted trapping efforts. I haven't really looked specifically at all the different reasons why they've been added. And for a lot of the specimens, there is some data missing. So, there were some specimens in Kentucky that weren't included in my maps because we didn't have county data. There are a lot of specimens that we don't necessarily have the specific data as to why they're collected. But from the ones that I did read, they did seem to be targeted trapping efforts. And then those specimens were deposited in the museum. So I think it is very likely that it is just management projects or theses that have been conducted. And this is the best way to kind of preserve those specimens at the end of the study.
Dr. Elliott, please chime in.
Yeah, basically the mammal collection, what you're dealing with, arose over many, many years of a gentleman who was here and taught mammalogy, Dr. P. Thompson. He had mammalogy, and then when I was hired, he eventually retired and I took mammalogy. But when he had mammalogy, the requirements of the students in the class for grade, part of their grade, they had to collect so many representatives of so many orders, so many families, so many species. And students being students, they would go, I think, and scatter and raid the surrounding Madison County. And that's kind of why you got such a hot spot on here. And then, oh yeah, I mean, we had to thin it out a little bit. At one point we had something like, I think 85 possums and things like that, because obviously that's a specimen everybody would get. And so you got a lot, but the reason you got your Madison sort of focused, and then you'll find the things from various other places where a student came from, by and large, where a student came from. You do have some from students that were doing research projects like part of MasterWorks, but your bulkier collection, and especially the unusual things like from out west and stuff, came from students who went spring break and brought things back. Because I used to talk to Pete when just before he retired about where it come from. And those were the stories that I got. I took over mammalogy and was going to continue with that. And then EKU started to get real serious about institutional animal care and use. And the people who were on the committee at the time, when it got started out, wanted to know why we had to kill these animals. Couldn't we catch them and look at them, or couldn't we look at pictures? And that kind of, I didn't want to fight that battle. And so I just said, fine, yeah, we can. And so things that have been added to the collection over time since then, as you noted, typically are related to a graduate student project or somebody doing, file something on the road, roadkill or something, brought it in. But by and large, you're Madison County strict because the huge bulk of it came from that one class, mammalogy.
Hey, Chuck, I'm wondering if you could share with us the possum races story from over in the old building.
The gentleman who taught mammalogy had some students that actually were founders of the EKU Wildlife Society. But we used to have annually in the old building, it had basically a rectangular shaped hallway that you go down one hall, turn right, come back around. Anyway, one of the annual events for mammalogy was students would go out and catch possums. And at night, when the security people weren't around, they'd bring them in and they'd let them go, and they'd bring them around the hallway of the Memor building, and the winner got a beer. It was known as the EKU.
How long ago was this, Chuck? I was never in on those. How long ago was this?
This was not to be advertised widely.
That sounds like a Bill Fair activity.
That would probably work pretty well around the balcony in the atrium. Wouldn't that be a good racetrack?
Oh my God.
Might lose a few down the stairs.
Might have a little trouble with animal care and use pity on that one.
Yeah, that would be one that would, be a flag, but it's interesting. You go back and look at some of those people and the name and stuff. They became directors of wildlife divisions and people directing wildlife agencies in different states. So we produced some unique people. But that was possum trot.
I just have a simple question with all this. How citizen scientists, how could we collect stuff to submit to all of these different collections? Or is that something that's open for local citizens to collect the mammals, the birds, whatever it might be?
I know we do have a collector's permit. I don't know exactly what states related to anybody picking up roadkill. And it's also important to note that, some species are protected. So you do want to be careful of the exact species that you are collecting if you're going to pick up species on the side of the road. But as Dr. Elliott mentioned, a lot of species end up in the collections because people find roadkill, and a lot of times they're still in good condition and you can preserve them. So I don't know exactly what our collection permit says, but I do know that we have one through the university. So if someone did find some roadkill, they might be able to kind of call and check if it would be okay to bring in.
Yeah, so far with me teaching mammalogy, it's been kind of an open door thing, and COVID put a damper on it this last time around, but I've started to bring back like a skin prep lab that we've done at least the semester, the iteration before, and that was a whole lot of fun. It was a big learning experience for me too. It made the building a little bit stinky for a couple of days, the zoo lab specifically. But that's something that, with having Sarah sort of working in the lab and kind of trying to get the mammalogy service position working on this kind of stuff, that's what I'm hoping we can, kind of reinvigorate the status of the collection. But it's weird, like I have a colleague of mine that's in Ohio, and apparently she teaches mammalogy also, and she has to have like a roadkill collection permit, but there's nothing that I'm aware of this explicit at KDFWR, and like I have my standard educational collection permit, and everybody seems to be on the up and up with how we're, basically just taking in that kind of, dead on the side of the road material that's so happily found for mammal museums.
And I think it varies by state, because I know at the University of Connecticut, we had a collector's permit that was pretty much anyone who found roadkill could just bring it into the museum.
Great discussion, folks. Any further comments or questions for Sarah or Luke or Dr. Elliott? Okay, Sarah, great job. I echo Dr. Elliott's applause there, and thank you all for coming. Next week, we will have 3 students presenting from my group, 2 undergraduates, Haley Bruss and Marie Baker, and one of our grad students, Miss Jane Dostart. So I'll see you all next week. We'll get an announcement out soon. And everyone take care, be safe, and we'll see you all around. Thanks again, Sarah, and thank you also, Shannon.
Thank you.